Hey, welcome back. Rick Dudley here, and we're going to continue our conversation on cytotoxic chemotherapy agents. Now, in the last video, we spent some time talking about anthracyclines, and we made mention of drugs like doxorubicin and daunorubicin. We talked about how these flat-looking molecules with a sugar residue attached to them have the ability to sort of intercalate DNA and interfere with, say, DNA and RNA synthesis. But we also talked about how these drugs have the ability to intercalate and bind to DNA that has been processed by that enzyme called topoisomerase 2, which is responsible for catalyzing the formation of double-strand breaks in the DNA for the purposes of synthesis. And when an anthracycline like doxorubicin binds to the DNA and the enzyme, it literally poisons the enzyme's ability to reseal those double strand breaks. What happens? You end up with an accumulation of those double strand breaks in a cell that's trying to proliferate. Consequently, you're going to get an apoptotic effect and hopefully cancer cell death. And we also made mention of some of the toxicities associated with the anthracyclines, cardiotoxicity being the big one. So, if your patient is a candidate for chemotherapy and the team is considering an anthracycline, you're going to get some baseline ejection fraction information on that patient to see where they are, how their heart is, such that we don't chemically induce heart failure with a drug like doxorubicin. Moving forward, next topic, we are going to talk about a large class of chemotherapy drugs that are collectively referred to as antimetabolites. And in this class are drugs like 5-fluorouracil or drugs like methotrexate or pemetrexed and a constellation of other molecules that we'll make mention of. Keeping with the same scheme objective-wise, I'll make mention of the pharmacophore where relevant. Talk a little bit about the mechanism of action for each of these individual molecules. And then we'll go ahead and talk about some structure activity if it's relevant and the toxicities associated there as well. Okay, now antimetabolites. When you think of an antimetabolite, really what I want you to envision is a molecule or a substance that serves as a false substrate in some type of important biological reaction. That biological reaction could represent the synthesis of DNA or RNA. It could represent the synthesis of the precursors for DNA or RNA. And when that antimetabolite, that false substrate, is present inside that cancer cell, it's going to inhibit the cell's ability to synthesize DNA or synthesize RNA. So naturally, then, what you would think is the antimetabolite drug is going to have some structural similarities to the normal DNA substrate or the RNA substrate, or even a cofactor for DNA or RNA synthesis. End result, again, slowing down or inhibiting proliferation, cell growth, and that can lead to then an anti-proliferative effect. Okay, these drugs that we'll make mention of in this class typically compete with binding sites and certain enzymes that are critical for forming those precursors, or we may find that these drugs actually get incorporated directly into the DNA or the RNA, or these drugs may compete with the enzymes that are responsible for synthesizing DNA or RNA. Now, clinically relevant antimetabolites include drugs that are categorized as folate antagonists. This includes methotrexate and another drug called pemetrexed. These are folate antagonists that interfere with the processing of folic acid, and we're going to spend some time revisiting that. We also have drugs that are referred to as purine analogs, cladribine and fludarabine. Now, these bicyclic purine base analogs uh, aren't used as often as some of the other drugs that we'll make mention of. So I'm not going to spend a lot of time talking about the purine analogs. On the other hand, the pyrimidine analogs, drugs like gemcitabine and cytarabine, and importantly, fluorouracil, are commonly used for a variety of different cancers, all the way from lung cancer to colon cancer to even certain types of leukemias. Now, first up, folate antagonist. Methotrexate ends up being sort of our prototypical folate antagonist. And if you think back to your classwork in immunology, when methotrexate was discussed as a drug for rheumatoid arthritis, it was a DMARD. Its mechanism of action is essentially the same. It's the dose that's really different here. Now, in rheumatoid arthritis, we use methotrexate to suppress the proliferation of immune cells that are causing the inflammation seen in rheumatoid arthritis. When we talk about methotrexate in an anti-cancer capacity, methotrexate is still going to inhibit dihydrofolate reductase, but its outcome is going to be to prevent the proliferation of not immune cells, but rather to inhibit the proliferation of cancer cells. Now, remember, dihydrofolate reductase is this enzyme in our cells that catalyzes reduction reactions. It reduces folic acid to dihydrofolate, and then dihydrofolate is going to get reduced down to tetrahydrofolate. All right, so we're successively adding hydrides essentially across the pyridine ring, and I'll show you what that looks like in a second. What's important though is once you get to tetrahydrofolate in this chemical pathway, tetrahydrofolate can get processed by another enzyme, ultimately leading to the formation of something called N5N10 methylene tetrahydrofolate. And it's this molecule that serves as a one carbon donor for thymidine synthesis. So this is what happens normally, right? Normally, tetrahydrofolate gets processed into N5N10 methylene tetrahydrofolate. And once you get to tetrahydrofolate, the N5N10 methylene version, this allows for the conversion of uridine to thymidine. So guys, it's reasonable to think if we shut down dihydrofolate reductase, we can suppress the formation of tetrahydrofolate. If we suppress tetrahydrofolate formation, then we aren't going to make N5N10 methylene tetrahydrofolate. And if we don't make N5N10 methylene tetrahydrofolate, we block this step in DNA precursor synthesis, and we induce thymidine starvation in our cancer cells. That will suppress DNA synthesis. All right, that was a lot to talk about. Now let's look at an image to help, again, reiterate the mechanism behind what's going on with methotrexate. Recall, folic acid gets taken up into cells through a folate receptor or a folate transporter. We have receptors on our cell surface that recognize folic acid and bring it into the cell. And recall also, folic acid is taken in by the diet. We don't make folic acid in our cells like bacteria do. Once folic acid makes its way into a cell through its receptor, it's going to get reduced. Dihydrofolate reductase or folate reductase will take folic acid and reduce it down once to something called dihydrofolate. So this bond essentially in the pyridine ring system is getting reduced. Once we arrive at dihydrofolate, again, dihydrofolate reductase will take that molecule as a substrate and reduce it down again, such that this double bond is now reduced. All right, so we've added a number of hydrides across the pyridine ring to arrive at something called tetrahydrofolate. Okay, Once we get to tetrahydrofolate, tetrahydrofolate will serve as a substrate for an enzyme called serine transhydroxymethylase. And serine transhydroxymethylase uses tetrahydrofolate as a substrate, and it essentially is bridging this nitrogen and this nitrogen in folic acid, right, with one carbon. And that's the carbon right there. And when that enzyme bridges those two nitrogens together, we end up with an intermediate that we call N5N10 methylene tetrahydrofolate. And this is the one carbon donor. That's the one carbon donor for thymidine synthesis. All right. Let's remove that and let's revisit this. But this time we're going to add on methotrexate. Here's methotrexate. Methotrexate has the ability to prevent these reduction reactions. And when methotrexate is present in a cell, methotrexate will bind to dihydrofolate reductase, and it will suppress the formation of tetrahydrofolate. We remove this intermediate from the equation. If we don't make tetrahydrofolate, we don't make N5N10 methylene tetrahydrofolate. And if we don't make N5N10 methylene tetrahydrofolate, we don't make thymidine. Okay? All right. Before we move on, I want to talk for a second about the image on the right. Folic acid, dihydrofolate reductase, and then another enzyme called thymidylate synthase are all sort of working in concert together to ensure that the cell has adequate thymidine concentrations for the purpose of DNA synthesis. Now, in the absence of methotrexate, right, when methotrexate isn't on board, we'll make N5N10 methylene tetrahydrofolate in adequate quantities. What this intermediate will do is it will actually work its way into the active site of this enzyme called thymidylate synthase. And along with N5N10 methylene tetrahydrofolate in the active site of thymidylate synthase is uridine. When these two substrates come together in the active site of thymidylate synthase, uridine, specifically the double bond between the 4 and the 5 position, will actually come up and attack that methylene carbon between N5 and N10 position in that methylene tetrahydrofolate. It will pull it off and remove it. And in doing so, you end up forming thymidine. And then thymidine goes into DNA synthesis. All right. So does this make sense? Do you see where interfering with the synthesis of tetrahydrofolate and subsequently preventing N5N10 methylene tetrahydrofolate can actually suppress thymidine synthesis, right? By simply denying the cell of this substrate, you can suppress thymidine synthesis. All right. As an aside, dihydrofolate reductase, again, it's using NADPH as a cofactor, as reducing equivalents. And inside the active site of dihydrofolate reductase, the substrate, folic acid, and the alignment then of NADPH has to be just right in order for this reduction reaction to occur. And it's set up beautifully in nature. Uh, Folic acid and NADPH, they line up very, very nicely in that active site to facilitate the reduction reaction. But what you're going to notice is that methotrexate, as a folate antagonist, has a little bit of a different binding scheme relative to folic acid in the active site of dihydrofolate reductase. And methotrexate is able to bind dihydrofolate reductase in the active site, but it doesn't get reduced because the electronics associated with the tyrosine ring cause it to bind in just a little bit of a different fashion, keeping it far enough away from the cofactor and preventing reduction reactions. So mechanistically, antifolate drugs like methotrexate are competitive inhibitors of dihydrofolate reductase. They will compete with folic acid. And when they win out, which they will, if it's a concentration dependent manner, they will deprive the cells of, importantly, thymidine and ultimately purines down the road as well. Thymidine starvation suppresses cell proliferation. And this is really what we're trying to get at with regard to treating cancers. Now, keep in mind, methotrexate is not smart enough to know to only enter into cancer cells. Methotrexate will enter into other cells, rapidly proliferating cells in the body that are non-cancerous. So right away, you can start to predict then some of the adverse effects one might expect with methotrexate use and even methotrexate high concentrations. Okay. On the top, what you see is a rendition of how dihydrofolate is bound, okay, in the active site of dihydrofolate reductase, right? So this is folic acid that has been reduced at least one time, right? And when it's bound, as shown on the screen, it is properly aligned such that NADPH can add hydrides across the double bond between the 5 and the 6 position and reduce that double bond to afford you with tetrahydrofolate. And there's an aspartic acid residue in the enzyme active site, aspartic acid 27, that's establishing sort of an important ionic interaction with the nitrogen at the 5 position in the tyrosine ring. Okay. When methotrexate is bound in the active site, what you really see is a little bit of a different chemical arrangement with methotrexate relative to folic acid. And the amino groups seen in the ring system end up causing this nitrogen that I've just enclosed in the box as the most basic position. The N1 position in the tyrosine ring system ends up being the most basic position in that particular ring system. Since it's most basic, it's going to grab onto a proton, bear an ionic charge, a positive charge, that is going to allow then for this ionic interaction between the same aspartic acid residue. But what you can see then is that all of a sudden its binding is going to be a little bit different in the enzyme active site. And as a result, we have too far of a gap between NADPH and the substrate. NADPH is unable to donate its hydrides across the double bonds in methotrexate because methotrexate has led to some misorientation in the active site, and things stop. Right? We stop reduction reactions. Right? In methotrexate, if it's bound in the active site, taking up space, that means that if there is folic acid in here, it's not going to be able to make its way in. Okay? So that's mechanistically how this drug works. And why can't I just advance? Okay. So meanwhile, while I try and advance the slide, and I'm going to actually close out of our slideshow, unfortunately, because iPad doesn't listen to me. So let's try and do this again from current. Okay. Structure activity wise, again, since methotrexate is a folic acid mimetic, you would expect it to have similar parts as to folic acid, and it absolutely does. In blue, what you'll see here is what we refer to as a tyrosine ring. Methotrexate has a tyrosine ring. Folic acid has a tyrosine ring. Remember that folic acid is made up of two other parts, para-aminobenzoic acid and then a glutamic acid residue. Shown in red here is the para-aminobenzoic acid moiety, and then shown in green is the glutamic acid or the glutamate residue. And the glutamate residue ends up being important for a couple of reasons. Number one, uh, it tends to lend to recognition by the folate carrier or the folate receptor on the cell surface. So that facilitates recognition, binding, and transport across the cell membrane. And then once folic acid and once methotrexate in particular gets taken into a cell, a cancer cell, what we see is that that glutamic acid residue has additional glutamic acid residues added to it such that it gets polyglutamated. And polyglutamation actually aids in not only its activity, but its retention inside a cancer cell. Remember, cancer cells overexpress a variety of pumps, P-glycoprotein and other ATP dependent transporters to pump chemotherapy out of a cell. When methotrexate gets polyglutamated, it helps to keep it inside the cell. Right? Toxicities. This should come as no surprise. Bone marrow suppression. Now, remember, the cells in your bone marrow are constantly turning over. They are an area of high proliferation, and a drug like methotrexate and other chemotherapy drugs don't differentiate between cancer cells and non-cancer cells that are rapidly turning over. So we expect bone marrow suppression to occur if we're not careful, in particular with our dose and our monitoring. Nausea, vomiting, diarrhea, abdominal distress, not surprising there. Stomatitis, or inflammation of the mucous membranes. Esophagitis and oral ulcerations, this is commonly reported with methotrexate. And then, excuse me, hepatotoxicity has been reported with methotrexate. And we also can see instances of something called renal tubular necrosis. And this is associated with high dose therapy. There are certain lymphomas that require high dose methotrexate. Diffuse large B cell lymphoma that's in the central nervous system requires really high doses of methotrexate to make sure that you get adequate drug in the CNS to kill the cancer. And if patients aren't excreting the methotrexate accordingly, it can accumulate in the tubules and lead to renal tubular necrosis. Not only can renal tubular necrosis occur, profound life-threatening bone marrow suppression can occur if patients are poor excreters of methotrexate. And when we have situations that present like that, we have to be ready to administer sort of an antidote or a rescue medication called leucovorin. And leucovorin represents then an antidote for methotrexate toxicity. Leucovorin itself is a reduced folic acid analog. This drug is already reduced. Okay, it's already equivalent to tetrahydrofolate. And as such, it can go on to N5 and 10 methylene tetrahydrofolate, which can then go on to help convert uridine to thymidine, which can then help to synthesize DNA. Since leucovorin is already reduced, this can help jumpstart or reconstitute the bone marrow and allow the bone marrow to start turning back over and producing cellular elements necessary for white blood cell production or megakaryocyte production. Okay. And again, since it's already reduced, it doesn't care if methotrexate's there. Methotrexate will prevent the folic acid that you take in from getting reduced, but methotrexate can't prevent the leucophorin from entering into this pathway. Okay. All right. That's all I have about methotrexate. I will make mention of one other drug that's commonly used. That's a folic acid antagonist, right? And the trade name is Alimta, okay? And the generic name for Alimta is pemetrexed. Pemetrexed is used quite often in non-small cell lung cancers. Okay? So we'll talk more about that later this semester. But just be aware that pemetrexed has a similar mechanism of action to methotrexate. Right? When we meet back, we will go ahead and talk about the next set of chemotherapies, and those are going to be the purine-derived antimetabolites. I'll talk to you in a minute. Hi, welcome back. Sorry about that long-winded discussion on methotrexate, but the drug is still widely used, as is its counterpart, pemetrexed, so it's worth it to spend a little bit of time talking about them. But moving forward, a couple lesser used molecules, we won't spend too much time on them, but they're purine antimetabolites, and we'll also throw in some pyrimidine antimetabolites here as well. Now, the purine analogs, of which there again is no shortage, include drugs like cladribine, clofarabine, and then fludarabine. And what you'll notice is that these particular agents are represented by a base, a purine base, and then they also have a unique sugar moiety attached to them. And it may or may not be a ribose, or it might be a derivative of ribose, an arabinose group, for example. Now, these nucleoside derivatives can be administered to a patient, sometimes orally, maybe intravenously, depending on the cancer and the drug. Regardless, once they're administered and circulating through the system, these drugs need to be taken up into the cancer cells, and they're done so by nucleoside transporters. Once inside the cell, then, through this nucleoside transporter, the drug will get phosphorylated a couple times to arrive at a triphosphate nucleotide. And then as the triphosphate, these drugs can actually get incorporated into a growing strand of DNA. And when they do so, they actually can interfere with the topology of DNA. They can cause instability in the double helix, and they can actually lead to the DNA strand coming apart and breaking apart. They can also competitively inhibit the polymerase enzyme. And if you have a high enough concentration of your purine nucleotide triphosphate analog, it will competitively inhibit DNA polymerase. Now, some of these drugs will have this era denoted in their name, like clofarabine, fludarabine, and then cytarabine. And really, all that is standing for is the positioning of an alcohol in the ribose moiety. So in the arabinose derivative, the alcohol at the 2 position is pointing up, whereby in ribose, the alcohol at the 2' position is pointing down. So this is the 2' position, 3' position. Okay, so this positioning of the alcohol sort of pointing up is not normal. And when nucleoside or nucleotide analogs that contain that era substituent get incorporated into DNA, the double helix becomes unstable, and that can interfere then with the stability. So this slide basically summarizes what I just said on the previous slide. After intracellular transport through those nucleoside transporters, and after phosphorylation reactions occur such that you arrive at the nucleotide triphosphate, these drugs can get incorporated into the DNA. And as a result, you can have DNA breakage, which hopefully will lead to our favorite cell death mechanism, apoptosis. They can also serve as false substrates, inhibiting DNA elongation by competitively inhibiting DNA polymerase. And we already mentioned strand breakage. Okay. Here is a brief example of some of the molecules that we're talking about. And the first one that I'll make mention of is fludarabine phosphate. So fludarabine, if you look at this particular base, what type of mimetic is fludarabine really trying to emulate? You know, it's a adenine mimetic. Cladribine also adenine. Clofarabine also adenine. So these are adenine derivatives or adenosine derivatives. Okay. And you'll notice too, in some of these derivatives, they have a halogen at the 2' position. So a fluorine or a chloride incorporated into the ring at the 2' position. And what that really does is that helps to prevent these molecules from being metabolized by inosine dehydrogenase. It protects the molecule from being converted from adenine to inosine. And then fludarabine at the 2' position has that alcohol sticking up. Clofarabine also at the 2' position has that fluorine, not an alcohol, but a fluorine sticking up. And then cladribine is a deoxynucleoside derivative. Right. What is ribonucleotide reductase? Are you familiar with that enzyme and its function? Ribonucleotide reductase, very briefly, I'm just going to abbreviate it. Ribonucleotide reductase is the enzyme that's responsible for taking ribose. So this is a base of your choice, adenine, guanine, what have you. Ribonucleotide reductase will take the sugar, the ribose, and remove the alcohol or dehydrate the alcohol at the 2' position. So this enzyme has a magic eraser that it can just run over the screen like this and remove the alcohol. Now you have a deoxyribose equivalent where there is nothing here at the 2' position. So this is how you make your substrates for DNA. And these, of course, are going to be your RNA substrates, the ones that have the ribose sugar. So ribonucleotide reductase is the enzyme that facilitates the conversion of ribose to deoxyribose nucleosides. So it's reasonable to think then that if you somehow can find an agent that blocks ribonucleotide reductase, you can decrease DNA synthesis as well. And it turns out that these agents may inhibit ribonucleotide reductase. In that same concept, we have other agents that are nucleoside in nature. The difference being that these two drugs are pyrimidine analogs. So we have something called cytarabine, which you'll see abbreviated from time to time, Ara-C. And then we have another drug called Gemzar or gemcitabine. And again, these are pyrimidine derivatives. Now, mechanistically, these two drugs, cytarabine and gemcitabine, will exert a similar mechanism that we just described for the purine derivatives. These drugs can actually get phosphorylated a couple times and then get incorporated into a growing strand of DNA, causing DNA breakage because, again, here is an unusual presentation of a ribose that DNA does not want to see or deal with, and that can lead to DNA breakage. Likewise, the fluorine, albeit not very large in van der Waal radii, still presents a problem, a conundrum, for the double helix, right? Also, competitively inhibiting DNA polymerase. Now, keep in mind, cytarabine and gemcitabine are both cytosine derivatives. Okay, so they can competitively inhibit DNA polymerase, and they are going to try and block or prevent cytosine or cytosine triphosphate from making its way into the active site of the polymerase. And if the concentration is high enough, these drugs will win out. Okay, and then I will make mention of one other interesting effect. Gemcitabine also inhibits ribonucleotide reductase, very similarly to what we just described for the purine derivatives. Okay. Toxicities. Where would we be without these toxicities? So these nucleoside analogs, these purine and pyrimidine derivatives, all can cause some type of leukopenia, thrombocytopenia, and other bone marrow toxicities. Not surprising. Nausea, vomiting, diarrhea. Again, not surprising. So those are the common toxicities associated with these nucleoside pyrimidine and purine analogs. I'm sure there are other adverse effects, but again, too, owing to the fact that the purines aren't used all that often, and we'll spend more time talking about drugs like cytarabine and gemcitabine later on, we won't spend too much more time on it. Okay. All right. What else do I want to say? I think that's all I'm going to really say about the purine and pyrimidine derivatives at this point in time. Next up is going to be a brief discussion on this particular slide. So I forgot about this one, you guys. I apologize. This sort of summarizes the activation process then for cytarabine or any other nucleoside that has an Ara substituent at the 2 position. Cytarabine or Ara-C is given intravenously. It can be given intravenously at a variety of different doses depending on its indication, right? Leukemias are a common indication for cytarabine. After it gets injected or infused into the patient, again, cytarabine will encounter these nucleoside transporters. These nucleoside transporters that are expressed on cancer cells are greedy. They want to take up as much nucleoside as possible so that the cell can proliferate. And this nucleoside transporter is happy to grab onto cytarabine and pump it directly into the cell. And once it's in the cell, that's when the kinase activity is going to occur at the 5' position on that arabinose ring, right? We're going to get the monophosphate, and then we're going to get the diphosphate and the triphosphate, right? And ultimately, we end up with aracytidine triphosphate or cytarabine triphosphate. And this is the active ingredient. This is the active moiety of the drug that competitively inhibits the polymerase or gets incorporated into the DNA to cause a cytotoxic effect. All right. Alternatively, there is one pathway that Ara-C can take. And if we allow this drug to have any affinity for deaminase enzymes, right? The deaminase enzyme can convert cytarabine into uracil arabnoside, which is otherwise inactive. Right? Do you see what's going on here? Up at the top, we have an amino group at the sixth position, and that's the difference between cytosine and then uridine, which has the carbonyl group up at the sixth position. A deaminase enzyme will convert the amine to the carbonyl group, and a deaminase enzyme will convert cytosine into uridine. And if that particular reaction occurs on our drug, we end up generating an inactive uridine derivative. Okay. So we don't want our cancer cells to overexpress these deaminase enzymes, but it would be nice if our non-cancerous normal cells had adequate expression of these deaminases to protect the cells from the toxic effects of a drug like Ara-C. Okay, now I'm done talking about nucleoside analogs. Next up, we will talk about a drug called 5-fluorouracil, which is very interesting, very complicated, but sees a lot of utility in the treatment of a variety of different cancers. We'll catch you guys in a little bit.